Yes, so I think a lot of questions in this direction are unresolved. That's why I'm doing research on that. And the tumor genetics makes new kinds of changes proteins, and these proteins can be recognized by the immune system. So, and this is, it could depend on what mutations you have, if the tumor can be recognized by the immune system. But the way around, a functional or non-functional immune system also may influence which kind of tumor cells can survive. So if the immune surveillance doesn't work, possibly a lot of different clones can appear and the immune system can no longer eliminate these clones. So I'm a bioinformatician, so I'm looking to this question, to this immuno-oncology question from the perspective of high throughput data like NGS data and gene expression data. And in the work I presented here, I analyzed data from the Cancer Genome Atlas, where we have all this data, but we also have H&E slides where pathologists can do quantification of tumour-infiltrating lymphocytes, TILs. I could reproduce the finding that in certain molecular subtypes of breast cancer, for example, the triple-negative breast cancer, you have more immune infiltrates. I can measure this by TILs, and I also can measure this by specific markers of mRNA, which can give me more specific information about different kinds of lymphocytes, like T cells, for example. And what I also studied is I looked if I have the TILs that are determined from the slides with which genes are correlating most with this. And the result was that typical T cells markers like CTLA4, for example, have a high correlation with the TILs. This is a contribution to understand more about the interaction between the genetics and the immune system, and to single out immunological hot tumors, and maybe to make a more quantitative or to look if there could be different kinds of lymphocyte. That's why I looked for T cells, B cells, and neutrophils and other kinds of immune cells in these tumors. And I hope this can contribute to a better selection of patients for immunotherapy. What will be next in my research? I would like to look if these markers I investigate here, if I can apply it in patients that are treated by immune checkpoint inhibitors, and if some of these markers are possibly predictive if this immune therapy helps. Okay. So I want to look in clinical trials where immune therapies are tested and do biomarker analysis in these trials.